Fatty acid metabolism by Mr. Ansari Altamash, Assistant Professor of Department of Pharmaceutical Chemistry, Y.B. Chavan College of Pharmacy, Aurangabad. Overview of fatty acid metabolism. It takes place in different parts of the body. The synthesis of lipoproteins, that is very low density lipoproteins, are synthesized in liver and they are transported to the adipose tissues in the form of triacylglycerols. Similarly, the synthesis of chylomicrons from dietary lipids takes place in small intestine. They are also transported to the adipose tissues, and these triacylglycerols are helpful for the synthesis of new fatty acids. Whatever fatty acid is synthesized by the body, they are utilized by muscles, livers, heart, and other energy production. Let's see the beta oxidation of fatty acids. Fatty acid oxidation or beta oxidation. The fatty acid oxidation takes place majorly by three steps. Number 1, the activation of fatty acid occurring in the cytosol. Number 2, transport of fatty acid into mitochondria. And last, beta oxidation in mitochondria, that is also known as proper beta oxidation process of fatty acids. Step 1, activation of fatty acid, which takes place in cytosol. The fatty acid is converted to acyl adenylate by the help of enzyme thiokinase. During this conversion, one ATP molecule is converted to AMP, and that AMP is added on your fatty acid molecule. In the next step, the acyl adenylate is converted to acyl coenzyme A by combining with the coenzyme A, and AMP will be removed. Step 2, transport of fatty acid into mitochondria. Acyl coenzyme A in cytosol is combined with carnitine by the help of carnitine acyl transferase 1, which will produce the acyl carnitine, and that is permeable to the mitochondrial membrane. Once this acyl carnitine is transported through the membrane, it will be available in mitochondria. In mitochondria, the second enzyme called carnitine acyl transferase 2 will detach the carnitine from acyl factor and gives you the acyl coenzyme A, which is the activated form of fatty acid. Third, beta oxidation proper in mitochondrial matrix. These beta oxidation steps are completed by 4 subreactions. Number 1, oxidation of acyl coenzyme A by the help of acyl coenzyme A dehydrogenase enzyme. Number 2, hydrolysis by enoyl coenzyme A hydratase. Number 3, oxidation by the help of enzyme beta hydroxy acyl coenzyme A dehydrogenase. And lastly, the cleavage of the beta hydroxy molecule, that is called beta keto acyl coenzyme A by the help of thiolase enzyme, which will give you the 2 carbon containing compound, that is acetyl coenzyme A. The overall reaction for one cycle of beta oxidation involves the substrate as acyl coenzyme A plus FAD plus NAD plus H2O and the coenzyme A, which will give you the formation of two carbon less compound, that is called acyl coenzyme A plus two carbon containing compound acetyl coenzyme A, which will enters into the Krebs cycle for the energy synthesis, and FADH2 and NADH2 molecule will enters into the ETC reaction for the process of formation of ATP. Energetics of beta oxidation. 7 cycles of beta oxidation produces 7 FADH2 molecule, which will lead to the formation of 14 ATP, and 7 NADH molecule, which will enters into the ETC cycle and gives the 21 ATP molecule. From 8 acetyl coenzyme A molecule, oxidation by citric acid cycle, each acetyl coenzyme A provides 12 ATP, that will multiply by 8, which will be 96 ATPs. The total energy from one molecule of palmitoyl coenzyme A, that is 131, but 2 ATP are utilized for the activation process. Hence, the net yield of the oxidation of one molecule of palmitate gives 129 ATPs. Ketone bodies. The compound, namely acetone, acetoacetate and beta hydroxybutyrates, are known as ketone bodies. Only the first two are the true ketones, while beta hydroxybutyrate does not possess a keto group. Ketone bodies are water soluble and energy yielding compounds. Acetone, however, is an exception since it cannot be metabolized. Ketogenesis, the synthesis of ketone bodies, which is occur in liver. The enzymes for ketone body synthesis are located in mitochondrial matrix. The substrate like acetyl coenzyme A, which is formed from the fatty acid oxidation, pyruvate from the TCA, and some of the amino acids are useful for the ketone body synthesis. Ketogenesis occurs through the following reactions. Step 1. 2 moles of acetyl coenzyme A condense to form the acetoacetyl coenzyme A. This reaction is catalyzed by thiolase enzyme, which involve the final step of fatty acid oxidation. Step 2. Acetoacetyl coenzyme A combines with the another molecule of acetyl coenzyme A to produce beta hydroxy beta methyl glutaryl coenzyme A. The HMG coenzyme A synthase is responsible for catalyzing this reaction, and this enzyme is going to regulate the process of ketone body synthesis. Step 3 is a simple process. In this, HMG coenzyme A lyase cleave the HMG coenzyme A molecule to produce two components. One is acetoacetate and second is acetyl coenzyme A. The newly formed acetoacetate spontaneously undergoes the decarboxylation process to form the acetone, which is a ketone body. Another enzyme, that is beta hydroxybutyrate dehydrogenase, converts the acetoacetate into beta hydroxybutyrate, which is also a ketone body. The overall process of ketogenesis, which starts from acetyl coenzyme A and ends at acetone and beta hydroxybutyrates. Lastly, we can see the summary of ketone body synthesis, their utilization and excretion from the body. They are synthesized in the liver, utilized in blood, transported to kidney, lungs, and excreted through the urine. Thank you so much for watching my video on fatty acid metabolism and ketone body synthesis. You can also subscribe my channel by Altamash Ansari.